Before we begin, I'd like to acknowledge the Wiradjuri people as we're joining from Orange, from our beautiful Charles Sturt campus in Orange today. So I'd like to acknowledge the Wiradjuri people as the traditional owners of the land on which we are meeting. And I'd like to recognize their unique contribution to our environmental and cultural heritage, past, present, and future. And I'd like to extend my deepest respect to all First Nations people here at Orange or online viewing this Provocations lecture. My name's Mark Evans, and I'm the Deputy Vice-Chancellor of Research here at Charles Sturt, and your host for this evening. And this event has both a face-to-face and online audience. So a very, very warm welcome to both audiences. Now we're going to be using Slido to elicit questions for our speaker. So please use this QR code in the frame behind me if you have a question. Now, Provocations is a series of public lectures and discussions and blogs written by prominent thinkers that seek to address some of the grand intellectual and social challenges confronting Australia and the world to enable progress. So I would once again like to take this opportunity to direct you to the Provocations blog at https://provocations.csu.edu.au, and you'll see this on the presentation slides later on. So there you'll find a series of blogs, series of provocations that are written by people who have presented lectures on this series, and you'll see that there's already quite a vibrant public debate happening around some of the key themes that are articulated in the lectures. So there you'll see blogs written by provocateurs Clive Hamilton, Sharon McLeod, Wayne Hudson, Lee Baumgartner, Garner Pogrebner, Patrick Walsh, and many others. And as I said, on each topic, we encourage reasoned online engagement and debate. Now, this evening's lecture is going to further strengthen our range of provocations. Tonight, Professor Mohammed Shiddiky, a relatively recent recruit to the Rural Health Research Unit here at Charles Sturt, but I'm sure we all know Professor Shiddiky. I have to say he's one of the most warmest and kind people I've met in my time here at Charles Sturt. He's going to be talking on the topic, keeping plants and humans healthy, on-site molecular testing methods and devices for healthcare and agriculture. Now, by way of introduction, Professor Shiddiky is a distinguished professor in nanotechnology in terms of how nanotechnology is applied to solving research problems, particularly in regional and rural New South Wales. But what is unique about Professor Shiddiky's work is the way in which those technologies can be applied to a whole range of different problems. He is a fellow of the Royal Society of Chemistry and a member of the Australian Research Council College of Experts. He's considered a pioneering figure in the development of biosensing methods and portable diagnostic devices for various biomedical, agricultural, and environmental applications, and is currently leading a team of researchers in this area. And before joining Charles Sturt, he held prominent positions at Griffith University, the University of Queensland, and Monash University. He's a prolific publisher in the leading journals in his field. He was the winner of the Griffith University Vice-Chancellor's Award in recognition of his outstanding contributions to research. And according to the 2023 Stanford rankings published last year, Professor Shiddiky is amongst the top 2% of all researchers in the world at the moment, so very heavily cited. He's also married with two beautiful young children aged 12 and 14. And look, we're very, very proud to host Professor Mohammed Shiddiky's research here at Charles Sturt. I give you Professor Mohammed Shiddiky.
Thank you. Thank you, Mark. Actually, Mark Evans said everything about me. I think he already said half of my talk, what I will be talking. Thank you very much, Mark, for a nice introduction. It's really so kind of you. And I'm very happy to see all the beautiful faces here, including our Honorable Vice-Chancellor. Thank you very much for attending this session, and I'm very honored to see you in here. And before going to talk about my some of the work I have been doing in couple of last almost a decade, and my team are running some of this project. I would like to just give a big acknowledgement to traditional custodian of the land on which we are meeting today, and pay my respect to the elders past, present, and emerging. And as you can see that Mark already introduced and said a little bit of my work, what I'll be talking today. Mainly, I'll be focusing on agricultural, diagnostic related some of the work which we have done. But a major part, I'll be talking about the human health related things. As you can see that in Australian government put a lot of attention to few of the areas. One of the areas is health. And my component, health means is big, right? It's not like only a simple health-related, detection, which I will be focusing on. I will be talking about some of the diagnostic devices that can be useful for detecting health-related problems. But this is the one of the major priority area for the Australian government. And government put a lot of this is one of the priority area in the sense is last year budget also they put a lot of attention to put this is the one of the area which ARC and NHMRC is putting the high attention. And I will be talking something related diseases that is called the cancer, infectious disease, some of the metabolic syndrome related problems I'll be showing in the coming slide. And second part, I would be highlighting agriculture research related area where I worked on the couple of project on couple of areas, but I think I will be giving one of the importance that this diagnostic devices, what we are developing, that could be used for solving many of the agricultural diagnostic related issues. Things. That means in other way, biomedical devices, what we are developing, same devices or almost similar technologies could be used for detecting the agriculture-related problem as well. Right? I'll be showing some of the example as well. And finally, in the last part, I'll be showing something government has, Professor René Leon and Mark Evans knows that government has a lot of initiative on this advanced manufacturing area. And I know this in the last year budget is the huge portion of the component government put in this particular area. Like they would like to see large scale manufacturing, facilities within Australian soil. And because this is the area where we are actually way, we are not even competitive with respect to the world, We are way down the track is very low compared to the other part of the world. But then our government is putting a lot of initiative in the advanced manufacturing area where we can set up the large-scale manufacturing industry. And in my research where in the agricultural side, and this in the biomedical device side, POC device or agricultural devices can be used, supplementary to this type of large-scale manufacturing site. So, in first part, when I will be talking about the health-related things, what I will be talking mainly of the effective technologies for health. So my area of interest would be a couple of disease here, which I mentioned. I'll be giving some of the impact, the first few slides, and to show you the what problem we are dealing with. So one of the major problems is the cancer. Second problem, I will be telling a little bit of the maternal health related issues. Thirdly, I will be talking a little bit of the metabolic syndrome. And if you can see, this cancer one is one of the major problem we know. Particularly, if you look at this in New South Wales, even 2024, look at this, 52,000 people are already diagnosed with the cancer. And the major all these diseases, cancer, we are dealing with this prostate, breast, melanoma, bowel, lung. But if you look at this slide here, this is the global burden of the female breast cancer. Look at this one from the socioeconomic index from the 0 to, if you look at this high income countries to the very low income countries, no matter, for some funny reason, middle income countries doesn't have just too many of the cancer incident. But in all these cases, with the time of 2010 to 2019, last 10 years, cancer actually is increasing. But we are expecting other way, right? It should be decreasing, the burden, but it's increasing. And you will see this is the example, many example is coming in the future also. But then this cancer is taking a lot of money. Look at the money, amount of the money it's taking. We are talking about the 25 trillion for the next 30 years. It's a big money. Now, it takes our life, it takes money. So that's the big problem for the cancer, right? Equally, if you look at the maternal health diseases, I'm interested in particularly these two particular diseases, gestational diabetes and preeclampsia. This also has the similar level of the problems. When it attached with all these, risk factors, and you can see, and I know the RSRI, many people are working on this, this gestational diabetes related problems. It's one of the big killer and a big problem, health related problem in maternal health. And when it comes to the preeclampsia, this is the almost 10% of pregnancy globally are affected with preeclampsia. And when it comes to the, look at all these risk factors, you'll see there are many people is affected with this one. And it can be one of the big problem, particularly in the maternal health. So that means this is equally, problematic for our life, particularly for our moms and upcoming babies. So this problem, what I said, is equally, if you see this global context here, look at this in Australia. In terms of these particular diseases in African countries, and India is one of the major problems, and we know the reason obviously. But then if you look at this on the gestational diabetes in the rich side also is one of the dominant, right? So the issue in Australia also, maternal health diseases like preeclampsia or preeclampsia is not maybe that high, but still we have almost 41,000 cases. There's no death, but there is a lot of cases, and the amount of the money government spent is severely high, right? So, and then other problem, if we talk about another angle of the problem, this is the metabolic syndrome related problem. The two particular disease I am interested, one is the non-alcoholic fatty liver disease, which is called the NAFLD, and other one is NASH, which is another term of the MASH recently, people say. This problem is way beyond even you think. Look at this on the problem, what you start is that we have a healthy liver. It gets start for some funny reason, or definitely some reason, it got the fibrosis in there. But on this fibrosis, actually with the time, it can be transferred to aggressive form, which goes in one stage is like a carcinoma. So when this healthy liver to go that stage is called the NAFLD. But NAFLD to all the way to go to carcinoma, this is called all these stages is the NASH. And that stage is irreversible. That means we never get back, like from healthy liver to NASH condition is reversible somehow. But the NASH to all the other two stages are irreversible. And we eventually, from that stage to up to that stage, up to that level, almost 10% people died. But 10% of total global population I'm talking. This is one of the big problem. Because look at this, all this is the risk factors, the type 2 diabetes, obesity, and all these problems are very common. Obesity is one of the major problem. We know that. So now look at this one. Once we go for healthy liver to the NASH liver, NASH to the this your carcinoma stage, but from here only one treatment, that is the liver transplant. But when you do the liver transplant, look at this one. Again, you are not getting back your healthy liver. You are getting your NASH liver. Even almost 100% cases it come to NASH. So you never get back your healthy liver. So this is something we would like to do so that we can develop a technology or method that can maybe guide, so we can identify which particular patient will go up to the NASH level. Maybe some of their lifestyle changes, some of their dietary changes, and that could maybe be a game changer for there. So, but then this burden, you can see this problem. But then look at this one, number-wise is almost 40% people who will be affected with this disease across the globe. In Australia also, within this 40% in next 10 years, you can see. But look at this one, this curve, slope is so high, right? And in case of the money-wise, it's taking your trillions again. We are not even, it's just, look at this one, how much of the money it's taking per year in European some countries, 35 billion they are using per year. So what I'm telling you in last few slides, so cancer is the big problem. Maternal health, also a problem for our moms and upcoming babies. And this one is the big silent killer. It is around us. Oh, we didn't notice this. And I would like to do some research where our technologies can actually solve this problem, or can we facilitate some of this diagnosis process and management process. So currently, most of these diagnostics method used for detecting all these major diseases I mentioned, these are the major things. Either imaging technique they use, or the molecular biology-based technique they use, or the tissue biopsy. Most standard one is the tissue biopsy. It's the invasive one, costly, and we know all these associated issues with that. But to be honest, not as of today even, all these methods are not suitable for the early detection. Because what we define as early, now question is how early is early. So how we define the early, this is not the suitable for the early detection. All these methods, we start to use all this methodology when we already have the disease. So that means when we have the disease, when I go to the doctor or I go to the surgeon, then only I know that I have the disease, which is I am already carrying. So it's stage 1 or stage 2. So it's not the early detection, right? So, but then according to the definition, importance of the early detection, look at this early detection importance. That's the only one slide which give you, there's so much hope that if we go for the early detection, in some of the cases, survival, is almost 100%. Ellen was actually debating with me this morning about the prostate cancer. But that's the data I have, collected. But it is, some of the disease, you can see this, it is true that the early detection is how useful is the green one. But when we go for the late detection, look at the survival rate. So that is clearly saying this in case of the cancer, but the most important things is not the data here. So important is this gentleman's statement. The Sydney Favor is telling that a method of detecting the presence of cancer before there are any clinical signs of symptom is the best solution for many of the diseases, particularly for the cancer. And that gentleman is considered to be the father of chemotherapy. We know the Dana-Farber Institute in Boston, after his name. So I think this is something telling the why we need early detection method, right? So now, when I got the early detection method, if you go, previously people in last 10, 15 years, people used to think about the assured method. But now after the COVID, they put the two reassured method. And then all this, this is actually well understood by the technologists who developed the diagnostic methodology. The reassured means real-time connectivity, easy of specimen collection. These two words they use, and then all the other things, you look at this, affordable, sensitive, specific, user-friendly, we know all these things. So a diagnostic method which we tell need to be used by you at home or personalized medicine, these are the criteria it should have. I'm not sure whether our device will have all these criteria or not, but we are working towards it. And I know that many of the things here, particularly sensitive, specific, user-friendly, rapid, and in some cases equipment-free, these are the criteria already within our device, activities. So this is the work I will be like. I spent almost 18 minutes on the introduction only, but I thought the reason I told all these things to give you a snapshot that you understand the burden and the problem of all these major diseases, right? So now these are the things what I will be doing in my lab and what we are doing in last couple of years. So these are the biomarkers. So these biomarkers means biological signature or biological molecules that gives us some identity of the presence of a disease. And when I define this one, starting from the microRNA to DNA methylation to cell-free DNA to circulating tumor DNA to here cluster DNA, cluster microRNA, whole lot of things. And my group from last couple of years are working on all these biomarkers. And how we are detecting? We are detecting two different ways we are detecting. Either we are developing a genetic, epigenetic screening-based method, or we are cellular or molecular biopsy-based technology. And in both the cases, we are either making the diagnostic device or diagnostic method. So now, I will be telling where we are doing this one, in the blood or saliva, or urine sample. So we are not doing in the tissue. So we are not invasively cutting anything. So we are collecting the blood, which is the minimally non-invasive. Urine, simply non-invasive. You can go to the washroom and collect the urine. Saliva, completely non-invasive. So essentially, you are using a non-invasive way of collecting your markers. That's why, and you are doing this on liquid. Essentially, you are repeating the biopsy. That's why we define this on liquid biopsy. So this liquid biopsy technology we are using, and this is the well-known term. One of the technology I would be like to sharing with you, which I have developed in a couple of years before. So that's the device, and I'm not going to explain all this engineering of this device inside, but essentially what we are doing, we apply the electric field in the device. And when the biological molecule are present in there, this biological molecule will start to flow between one side of the device to the other side of the device. When all this flow is going on, there's interesting things is happening within the device. That's actually happening here. So what is, device is coated with the antibody that's specific to your target. Here our target is the red monster. This is the cancer cell. So now this cancer cell is specific to some of the antibody is printed on the device. When the sample is passing through and they have the specific interaction with the cancer cell, they get caught. But the question is, why the other things are not catching on the surface or got absorbed on the surface? Because we apply the electric field in such a way they can share off all the non-specific one, and it can only catch the specific one. That's why the device name is nano-sharing device. It is a share away, kicking out from the surface. So this one is just like another one we built like a airport security gate, almost the same technology. What we have done is all these pillars are sitting like a airport security gate, and all these terrorists are going with the red one. They cannot pass through, because if the one they pass through, the gate will beep beep. They got caught. This is exactly how we got caught them on the sensor surface, and all the unwanted one is passing by. Right? This is the one where, when if you are working in the sensory area, you know the circulating tumor cells are extremely rare. That means they are present in 10 or 20 in 5 ml of blood. So you need a methodology where a 1 billionth of the unwanted things, and you have only 10 wanted. So it's like a 10 red shirt you are putting in China, in China, like 1.4 or 1.5 billion people. 10 people you put with the red shirt, and all other people are white shirt. So now you need to catch this 10 out of this 1.47 billion. So you need a technology that is extremely specific and highly sensitive. So I think this is the technology got highly high potential in there. And I just saw with the time, I don't want to go the whole picture in there. So circulating tumor cell, I just show the example. But this technology can be used for CTC cluster, for the extracellular vesicles, which I will be coming in few slides later. This is another vesicles, which is like extracellular matrix related biological entities. Protein biomarkers, any proteomics things, you can catch this, and the pathogenesis, particularly for the parasites. And you can catch them. You just need a specific antibody on the surface, and you can catch them. And all the unwanted one will go out. And you can tune up and down depending on what you want. So it's tunable. So that's the wonderful technology actually in there. So another one which we define as interfacial biosensing. By the name, you know the definition. So it is something going on the interface. So what is that then? So this is like you have a molecule, healthy, it's a compound collected from the healthy patient or healthy sample. You have another sample collected from the cancerous sample or disease sample. Because I'm a chemist, right? I define in chemist way. Let's say it's collect from the sample that is not okay, and the sample that is okay. So now I have two different samples. Do you think both these two different samples will be absorbed on the surface similarly? I don't think so. They will be absorbed differently. And when I learned that one, then I realized, if this is the case, crazy hell, why don't I just use that differential affinity as a method of detecting cancer or detecting some disease presence? That is called the interfacial biosensing. And it comes from that idea, simple idea. What we have done, DNA methylation. For the people who don't know what is the DNA methylation, is DNA, is cytosine is there. This cytosine somehow got crazy and got the methyl cytosine. So this is another new entity came, methyl group. So you have now two different compounds. One is methyl, one is the methyl cytosine. So the cytosine and methyl cytosine. Two will be absorbing on the surface differently. They will not absorbing the same way. And that's exactly what we have detected here using the breast cancer sample, like directly collected from the breast cancer, putting on the electro surface, and we have seen the two different level of the absorption pattern. And we tested this one in the large cohort of the minimal, not so large. It's around 100% sample are there, and it can be detected actually. It can be used for detecting the breast cancer. And that was the wonderful technology. This technology got picked up by, sorry about that. Can I have some water, please? I'm very sorry. Maybe I'm talking too loud. But so this picked up by Guardian. And then after then, as you know, as usual, Australia-wide, all these things taking up. I'm just showing the importance of this technology. And finally, it took by CNN also. And at the end, we got also the cover by the Courier Mail saying that it's a game changer. I think it was a game changer. It's based on that technology. There are the two Brisbane-based company. So with this technology, have another advantage, another side. So that side, you'll be loving that. So you see this, I know a couple of biologists here. This is like a prostate, it's a fusion gene. Normally, prostate cancer based on the prostate-specific antigen, PSA. I think everyone knows the problem associated with the PSA marker, right? I'm not going to repeat this one. PSA marker as a prostate cancer marker is a problem. So now we need alternative test. We know that if this particular this gene is present, that giving a signature of the malignant, but it is absent, is called the benign. So that means if you go to the test now, you have a lot of PSA, but that doesn't mean that you have a cancer. You need to check whether they are benign or malignant. And that test, we use the same methodology. Why I'm saying this one, this is interesting. So you collect a little bit of the technology, but I will not go for in details, just giving you a little bit snapshot. Collect the urine sample. From the urine sample, we are looking for a particular monster. That monster is, let's say, this black red one. But we need to fish it out, because there are the millions of the other things are present in there. We need to fish it out in the junk of molecules, right? How we did? We just used the molecular one probe, that's called the capture probe, magnetically triggered there. So we just magnetically separated them. I'm not going to explain all these things. But at the end, essentially we get the things what we are looking for. Once we get them, this thing we put on the electrode surface, and we know there is the affinity adsorption based on the previous work I have showed that there is the affinity interaction adsorption. And based on this one, we know that there's a bare electrode and cancer electrode. There is a two different response. It's wonderful. So this way, we can easily pick up which is the presence of the cancer in there, right? But interesting thing is not that. Actually, this has been tested also, the patient sample. You can see all this healthy one is a very low response, and the different stage of the cancer one has a different responses. It's wonderful. But then this is not actually the selling point. Selling point is that technology is not limited to only this prostate cancer particular sequence I just showed. It is a range of any other target you say, cfDNA, ctDNA, microRNA, mRNA, long non coding RNA, anything, any hell you tell, everything you can detect, using this one. What you need? Only a specific capture probe and a little bit of the modification of the method, depending on the sample type. For example, urine sample, saliva sample, and in the blood sample, you will not be dealing with all these three kinds of sample in the same way. A little bit of the modification in the early processing, but the end of the detection type would be the same. And that's actually becoming an universal method for detecting biomarkers of this type. And the importance and the technological barrier and challenges I have addressed in this invited 3 review paper, which is published in by top quality. If you are in this field, these are one of the top, all 3 of the top quality review paper published just recently on explaining all these fundamentals. If you would like to go, you can just have a look. Now, I would like to show something else now. So these are the device. One of my colleague, Amandeep Singh, is working on for many years on this. And we have developed something in there. So the device Amandeep and his team, or we all are working on, this is the device we build. And hardware to software, everything is built at home. So that means this is the machine. When you buy a machine of $50,000, $200,000, $300,000 machine, this is similar one machine we built. It does the same job, right? So this is, I think next slide will give you this device here. You see so many components in there. It's just starting from the power source to the display sources, everything in there. But then I think this is now how you look. Finally, you look like a small package. Yeah, this is the things you look. That's exactly what is this after the 3D printed device. And it has a multiple channel or multiple reservoir where you can do the multiple target detection in one go. Right? And it also has a display unit where you can touch screen so that you can, and because some people are colorblind. So if you get the white color, red color, blue color, they will say all colors are same. So you need a digital number. So that's why we need a touchscreen based digital readout, right? So these are the device. Now who will be using and where we will be using this device? These are the device where this bunch of people will be using in my lab. and they are using. So Fatima and Sharmin, Milkias, and Nazia, they are using two particular diseases. One is preeclampsia, which I explained earlier, the impact, and another is the NAFLD, which is the NASH. And how they will be using these devices because they also find that exosome, this is another new level of the biomarker. Exosome can be used as a biomarker for identifying all these diseases, particularly these two diseases as an example. So how they will be doing this exosome particularly has a bunch of biomarker. It is a cargo. It's carrying it's everything in there: DNA, RNA, microRNA, and also surface has lot of protein-based signature. Some of the example is some microRNA is mRNA is there. This case one, SIRT. These two are the Nazia is particularly interested in, because these two mRNA we can use Amandeep device for detecting these two, because these are a little bit longer micro mRNA, which we can use an amplification technique, which is the isothermal based. And that's exactly Nazia is doing, like collecting the plasma sample there and magnetically isolating this exosome initially. Once they get exosome isolated, and this isolated exosome, she will be just only blasting that, means this heat lysing. And after lysing the entire crude sample, she will be doing this isothermal amplification based one method which Amandeep device will be using here. And she is doing that. But now here one more thing is that particular part, this Fatima has done his PhD with me at Griffith and one of the university in Bangladesh. So she has isolated this exosome magnetically from there. So this magnetically isolated exosome, once we get magnetically isolated exosome, these things will be lysed in there and using Amandeep devices for detecting or for doing the rest of the analysis. It's fantastic, actually. Two-step process: exosome isolation and then exosome-based biomarker, which is actually this mRNA-based detection. mRNA-based biomarker will be detected. So this one, Milkias will be using the same device, and he is using this device for doing for NAFLD and for doing the NAFLD detection and giving the prediction for the NASH. I'll need another 5 minutes. Yeah. NASH detection, and this will be actually one of the technology, one of the way of detecting this all this biomarker. You can see there he can use this one exactly identical manner like the way Nazia did there. And because this is the platform technology, this device can be used in either cases. Now, one of my other student at Griffith is Sajidul, is doing wonderful work in there using the point mutation. And funny thing is, there also he is using the isothermal amplification method. And we have a couple of works done on the colon cancer and other cancer and also metabolic diseases. And Sajidul, this work will be implemented with this Amandeep device, and we are doing this one. Milkias is doing this one for detecting the point mutation. Fatima is working on this one on the point mutation detection. So bottom line is this device is a platform device for range of disease detection. So I would be like to next 5 minutes on the agriculture. I think I can finish it, okay? This is actually novel technologies for enhanced food production. So when we get, I need to show that slide actually. This is actually hired from Jeff Gore, my collaborator, and Jeff is present here. So these are the things you can see in last 50 years, next 50 years, the way it's going on now, the world. All these red places will be having the severe food crisis. Almost 60% of the world population, almost 70% of the world population probably live in this region. Australia also, maybe we will be producing a lot of in this side, but then other side is also starving. So, but overall, this is the problem, big problem. Maybe my child or my grandchild will be coming to this world with a lot of difficulty, this level of the problem. So one of the things, I cannot solve this problem, but maybe we can do on-site some rapid testing or handheld device-based some work where we can guide maybe with the help of Jeff and people like, expert like them. So the guide them, maybe the farmer, so can actually save a little bit of the, crops, crops, what is this called? Crops disease-related issues and increase the production. Quality of the food we can improve. Production we can improve. If we can do the rapid on-site detection, maybe can solving a lot of problems. So I'll show you how we can do. This is traditionally, people get on field, they get the sample, they send this one to the laboratory. You know how long that take? About a week. And these guys do the analysis in there, all this molecular analysis. And they send the data by not email, by post. In there, this age event. And that takes another 7 days. By then, crop's gone. Another crop start again. So this problem, the related problem is also there. Maybe Jeff knows and we all knows. These are in the molecular testing, all this testing they do, commercial kit-based analysis, multi-step process. We need to extraction the DNA, do these, that, whole lot of things. A lot of money involved. $500 to $700 only a kit. So I was thinking, okay, this is the wish list for diagnostic test. Do a simple test, less than $1, do it 5 times, do it 20 times, $20 cost, within 1 hour. You don't need to send this one to South Australia, Saudi. You can do on spot. So this one may be the wish list for us on field. We don't need the high accuracy like that test. But we can do 20 times and get the 18 times correct data. $18 cost maybe. So this way, I thought 2 considerations is needed. One thing is we need to simplify the method. Another thing is we need a on-site deployable test. And how we are doing this one, I'm just going quickly. On one of the plant pathogen, like this one is the sugarcane, one of the pathogen, Lipsonia, which we get from the xylem sap sample, directly hit this and detect on spot. We don't need DNA extraction, nothing in there. And this one, when we go for the leaf and menstrual tissue, it works exactly identical manner. We just need to soak this leaf sample and the tissue sample in the water, and water can be used for this, amplification. And Amandeep device type of that device can be used. That's the device we used for detecting this. This is also another handheld device made by one of our students at Griffith. And this device is essentially what you do: collect the sample, put in this little pen drop, and put in the device, and start on, get the data. And I think this is filling up some of the wish list in there. So with that, I thought maybe I have summarized a little bit, or I have shown some of the demonstration that we can develop, nanosharing technology can be a solution for the many problems in the conventional biosensing. And we have developed an interfacial biosensing method, which is a platform for many of the genetic testing related biosensing issues also, particularly for the genomics, transcriptomics, and proteomic analysis can be used. And exosome can be used for maternal health and also particularly in the metabolic syndrome, in particularly NASH or NAFLD. And I think maybe I have shown, demonstrate 2 devices that can be used for detecting many of the nucleic acid related biomarker and that target many diseases. With that, I must be saying this: all these beautiful people are currently working with me, and all the people at RSRI who is supporting me, particularly Alan Rose was a wonderful, and he's a good mentor, good friend, and good colleague to work with. And these are the people previously worked with me. My group at Griffith, which I miss them a lot. And thank you very much for everybody for listening. And Mark, I'm very sorry I took 4 minutes extra, as I promised.
Thank you, Professor Shiddiky. I mean, look, your passion for your subject is palpable. But as a lowly social scientist, through the questions, maybe help me do some sense making.
Yes.
All right. So the first observation I would have really, in relation to certain of these diagnostics, is that one of the significant benefits is obviously early detection, but also a lot of these diagnostics aren't as invasive as traditional methods. So for example, it's better for patient well-being than has been the case previously.
Yeah.
So that's a major stride forward. All right. Yeah, so we're talking really about understanding the benefits of the technologies. Clearly, there's a benefit around patient well-being in terms of the non-invasiveness of certain of these diagnostics. Then, of course, there's the issue of cost. So you seem to be saying that actually also traditional methods in these areas are extremely costly. Yeah. There's very, very intensive processes that underpin the traditional detection methodologies. Yeah. And this makes your technologies more affordable, not just to health maintenance organisations in regional New South Wales, but potentially globally.
Yeah.
Is that the case?
Yes.
Maybe you could expand on that a little bit
for
more.
example, let's say if we go for the cancer, right, as an example, like you raised it, I should be clarifying these things. But if you go for the cancer, particularly for the cancer diagnosis in one stage, let's say if we go for this traditional cancer detection, like if you go for, this is called the cell source method for looking for particular cells that related to cancer. So for doing this one, even though that method is now a lot of question though, but that method will charge you a couple of $1,000, and then time is around for about a week to get the outcomes, right? So, but then imagine that you have now time is involved, cost is involved, and then you need expert people to run interpretation and a whole lot of other things. But then if you have a non-invasive test, which may be not that 100% accurate, maybe 95%, maybe 85%, but you can do it personalized, like you are detecting like your COVID-19 or things like that. So in that case, even test device is less than $10, for example, let's say. So now if you run 20 times even, it's not even touching that cost. But then if out of 20 times, if 15 times come, okay, then you have something. Something needs to be considered.
Yeah. So this is a huge sort of method of inclusion in terms of combating diseases where time is critical. So look, we've got lots of questions now coming online. First question, when you talk about real-time data, what is the information and communication technique? IoT-based or Wi-Fi-based?
Okay. This is the something definitely. So when we go for the in-field, detecting some of the signature on-field, that data can be passed through or that's sent through the Wi-Fi or this, there's Amandeep is there, he can even help. But that things is okay. But when I say the real time, that means in the method analytical real time. So when the process is going on, the interaction between this target and our probes, what is going on, we would like to detect them in real time. So then we know the virus or fungal or pathogen loading on in real time. That's I mean by this. But yes, once we get the data, this data can be transferred to the other side of the world or other side of the country or a centralized lab by the Wi-Fi or other money is there for helping the computer science related things. That can be done. Yes.
Yeah, but it's dependent on other technologies.
Yes.
Yeah. Jeff Gurr asks, please tell us a little bit about the commercialization pathway, how to find industry partners, manage IP and bring to market.
Thank you, Jeff. Yes, some of the things what I just explained is already with the collaboration of some of the industry. That's why I didn't go to the whole details. The one I can tell, I told, right? Yes, it can be. But for example, if a new things comes, which I and Amandeep and some of my students and postdocs are in the process, where once it comes, we will follow the standard path in university and the way we do, and it can be. But yes, there are many people are interested. For example, I was approached approached by what is this called? New South Wales milk industry, and on last week, and they're asking me, they are detecting some of their pathogen in milk using the ELISA, and they would like to have a rapid test, which is inexpensive and quickly can be done, not like 1 day or 2 day, and like so they would like to put the money so that we can go for a linkage grant. So these type of things can be done. Yeah.
Another question. So obviously we need to wrap a commercialization network around you to actually help support that type of work. Something for Nerdosh to think about with Nick Paget. Another question, I see that some of these technologies are platforms for detecting biomolecules and can be used for humans and agricultural diseases. Can your technology be used to detect diseases in humans, animals, plants, and their environment in a One Health approach?
Yeah, thank you. Thank you, Mark. I was expecting that. I think, yes, I showed that some of the things, I think I will be going back if I can. Can I go back? Only one slide that will give you, I don't know which one. So if you look at this slide here, this slide, sorry about that, because this is important. This slide here, look at this. I am targeting one of the mRNA using a capture probe, right? So now imagine this mRNA or this sample is not the patient sample. It's maybe the plant sample, which we have done, right? But this sample could be a animal health related sample. We will be doing the same way isolation of the DNA or mRNA or microRNA. We will be using the same path. So I think essentially this technology a platform forms targeting from the human, plant, or even the animal. So it can be done, yes. And the same way like in this nano-sharing technology which I showed, the pathogen as targeting here, but then we also use the same technology for Giardia, and we also use the same one for Cryptosporidium. And I know this technology is working, and we have a bunch of data in there. So that means it's a bunch of what is this called field. We can use the same technology. Yeah, it's the platform.
So it's kind of almost an alternative to eDNA in that sense.
Yes.
Many people fail to develop anti-cancer drugs and diagnosis technologies. What makes your technology especially promising to resolve this problem?
I think the anti-cancer drug-related things, I cannot make any comment, but diagnostic things, one of the major problem in the diagnostics, why it's not working. For example, Oxford Biosensor, if I talk loudly, they will, maybe it's also public. But there are a lot of issues with the diagnostic technology development. The issues are two major problems. One problem is many people doesn't know how to avoid the non-specific absorption. Another way, false positive response. And that's the one. So if you know a little bit of the biology and chemistry because you need chemistry and physics to deal with the technology. So you need to also working on the interface where this molecular absorbing or detecting or the method you are using. Ideally, the answer to this question is diagnostic technology is, it's not true that the fail, because there are already many diagnostic technology out there which is accurate, and many upcoming technology also coming accurate. So it's just a way, if you understand better the chemistry, physics, and biology all together, I don't believe the diagnostic technology is a problem. But drug-related things, I cannot make any comment.
Now, if anybody has Any questions from the floor? Here, there's a question here.
Thank you very much, Shiddiky. Superb lecture, very enjoyable indeed. Just very quickly, so what you're doing essentially is you're picking up early diagnosis. That's what it is for the disease. If you're talking about breast cancer, for example, you have that wonderful method for detecting very early onset of the disease. But then once that's diagnosed as such, then the patient will go to the clinic, get a proper diagnosis, and then continue on with treatment. So essentially what's happening is that you're doing a pre-screen, as it were, of what would be a normal diagnosis. So in that sense, I guess you make it much easier for the disease to be treated because it's an earlier diagnosis. That would be the key advantage.
So, thank you for that. You picked up quite right, but at the same time, these technologies can be used for screening. That's true. But screening also economically very useful. Once we know, for example, regional New South Wales, site. So once we know this particular patient has some health-related issue, then that particular person needs to be treated properly. So this way, proper diagnosis, proper screening can help to get the proper treatment. At the same time, also possibly we can, let's say, we can actually reduce the cost, because then every person don't need to go to the high level of the screening and high level of the things. But that's the different level of the discussion. But at the same time, it can also help or guide the management of the diseases, because its early detection doesn't make any sense unless we know what to do with this, right? Yeah.
So a related observation, which kind of runs the counterfactual from Anne Johnson, has there been discussion about the risk of over-diagnosis and intervention when none is needed?
Yeah, it is the cases of many times. Like this is why this type of technology can go for one disease detection with the multiple biomarker. Instead of going for the one disease in the one biomarker, you can go for the multiple biomarkers saying the same story. So this way you can maybe potentially avoid the overdiagnosis. But for example, prostate cancer related things, the overdiagnosis is one of the known problem. We know that. But this type of inexpensive technology, rapid technology, can be used to reduce this overdiagnosis.
So a question really around the margin of error. When you mention 90%, 80% accurate, is it percentage accurate for individual patients or for groups or cohorts?
Yeah, it's like a diagnostics accuracy, right? It's like you run a cohort of the patient and see the data, and you can validate this one with the existing gold standard method. And relative analysis can give you this percentage information. But again, statistician here can give us the more information. But I do believe this information, what I was telling, is the cohort base. Yeah.
Right. Is there any comparison of biomarkers of early cancer screening using POC versus the lab-based methods? Or is that for physician use only as interpretation is needed?
No, there are already a POC test is available. For example, I don't know, is it cancer related? If not cancer, there are many. For example, this COVID-19 rat test is a POC test, right? So we know this is quite accurate also, right? Rat test is, rat test, right? Rapid antigen test. So everybody can run it now. Even my 12 years old also used on other day, and he showed me the photo and said, "Dad, I don't have the COVID." So I think it's quite useful, and it can be used. So the POC, this is one of the POC tests. Yeah.
So there's another question here about IP, which I know is kind of quite a contested question across researchers, not just in this university, but across Australia, which is about, well, what is your IP position and plans to commercialize this to make these great services and products for our community and people? So obviously the sort of contestation is between those who say the Shiddiky, you should have IP, in relation to all of these diagnostics, potentially, through a sharing arrangement with your team, and those who say, well, actually, no, because you're not going to get anybody to invest in taking the device to market if you do it that way. And what needs to happen is you need to be supported and your team needs to be supported to enable you to do long-term research and discovery. And that way you're more likely to attract the investment of industry. So where do you sit on that thorny question?
To be honest here, most of the things what I just explored today, I don't worry about any of the IP things because most of these things are published by my group, so it's already out there. So if anyone wants to use this, they can use. But the thing is, there are few things, know-how, which we know how to make it happen. So these are maybe the things which we are proud of, and we will be exploring that. Definitely, if anyone would like to discuss about this with us, I'll be most welcoming and talking with them, and then see how we can work together and find things moving forward and beneficial from both sides. Yeah.
So two questions to go, and then we'll probably be running out of time. But obviously, there's a very significant sort of ethical dimension to this. Obviously, once patients receive news about the issue, but the support systems that are necessary to provide them with the right sources of support moving forward. But I guess that isn't you, is it?
No, listen, whenever we do any of the research like this to go for a large cohort of the testing or validating this one, we do a proper ethical approval and proper ethics done. And then only we move forward. But to be honest, in last 15 years, I never get any challenge for getting this approval, and people usually very happily get involved and support us always. So I don't believe anything we have a problem. But most of the time, what we do, we work collaboratively with the clinician and surgeon and health professionals, and they get heavily involved. So we need to have the ethics from the both sides, from our university side and their sides, and all the things go for like the paperwork. So I don't believe it would be an issue, and it never been an issue with me at least. Yeah.
So final question: what are the next steps for your research?
Oh, that's interesting.
And just focus on the medical areas, the health areas.
So I think that my next vision would be like helping here 8 or 9 young people are working with me to making them happy with their research and make some of the device technology commercialize and see whether we can use and reducing some of the health burden and also the problem associated with the agriculture. Yeah, so it's a long vision. This is why I moved from Griffith to CSU, and thank you everybody to support me, particularly Alan and you and Rene, and that's what the vision is like to move next few years and see where we can see this application. But definitely, this all 8, 9 young people are working with me to make them happy and also build their career on this diagnostic world. And I know Australian government has a massive interest on advanced manufacturing, and that is associated with the plant, agriculture, and One Health, as you know. So all this is going to the advanced manufacturing side, particularly in Brisbane. I know that a few months before they put a lot of investment in New South Wales government, also doing the same. I do believe that this is the area I would like to work and see where the future goes. But definitely, first thing is we would like to see this one in real some outcomes. Yes, Ella?
Oh, about your conference coming up.
Oh, yes. Thank you for reminding me. So I'm running a conference from the 8th of the July to the 10th of the July. And our Honorable Vice-Chancellor, Professor Anna Leon, will be opening the conference. And this is the Australia and New Zealand Micro Nanofluidic Conferences. even though it's a fluidic, but we will be talking a lot of this type of technology which I presented today. This is the primary device technology people use or make or develop for detecting the diseases across the field. And there are many people are coming and some high profile. We have 4 laureate fellow is coming, and multiple NACMRC L3 fellows are coming here, and many distinguished invited keynote speaker also attending. We have close to 100 attending now, and it will be maybe going up, and in a couple of, still 2 weeks to go. I never expected that. I thought maybe 80, 90 people would be there, because last year was 90 in Adelaide, in the Adelaide Oval, and in Warrens is over 100. We are beating them. So I was so very happy. But it's all credit goes to my team. They did a lot of hard work, and support from Mark and Alan supported hugely the conference. Yeah. And I will be requesting everybody to attend just to see at least what we are doing. Yeah.
Well, that's fantastic. Getting a major conference here in Orange is absolutely what we want. We want to be at the centre of discovery in this area. Thank you so much for sharing your pioneering work with us here this evening. As Professor Shiddiky said, many thanks to Professor Leon for the tremendous support she provides to the research in not just in this area, but across the health areas more broadly. Many thanks to Nilma Matai and our production team for helping put this event together and to beam this lecture and discussion across New South Wales and beyond. We hope you will all join us for our next provocation lecture on Wednesday, the 28th of August at the Wagga Wagga Playhouse, when Professor Leslie Weston will address the topic, Adapting to Change, Invasive Plants and Pests Take Up the Challenge. See you then.
Thank you. Thank you very much.